So thank you very much to the organizer for the invitation. It's a great meeting. Congratulations, Rick. These are my disclosure. Well, I think that all people working with OCT-A nowadays are realizing that the most important improvement that we have with this technology is related to the possibility to have a kind of pseudo 3D visualization of retinal and choroidal vessels in normal and diseased eyes. And for the first time, we had the chance with OCT-A to look at, in a separate way, to the superficial, deep capillary plexus, choriocapillaries maybe, or choroid, and also to a possible intermediate layer between the superficial and deep capillary plexus. Well, this is something very helpful and useful, and it's something that I'm still using every day in my clinical practice on a qualitative point of view. The problem is that, as also Florence just showed us, quantification is extremely important in order to have a wider clinical and application and also repeatability of this application. So there are this question, are those quantification that we can do in a more standardized way really reliable? Should we trust in them? This is the first study in that kind. So the authors investigated the size of FIZ as well as vessel densities in two different cubes of visualizations of the 3x3 and the 6x6. And actually, using a spectral domain OCT that we know has some less potential resolution for deeper layers and including choriocapillaries, the authors realized that visualization of, analysis of FIZ is quite reliable, even if we compare two different sites and acquisition. On the other hand, this is not the case for vessel densities. So based on these first results, we wanted to assess with a higher resolution, with a deeper performing OCT, with a wet source OCT, this characteristic in this paper just was published this month in IOVS. And we investigated both normal and diseased eyes, so normal eyes and eyes of patients with diabetic retinopathy, so diabetic patients, 3 different readers and 3 different scan patterns, so 3x3, 6x6, and 12x12. And this is the way the study was carried on. So we first looked at FIZ, and then we looked at the 3x3 areas by cropping the 3x3 areas, as you can see here, on the 6x6 and on the 12x12. And all images were then actually binarized in order to have the 3 images comparable. Of course, these are the demographic characteristics of the population that were quite similar, except for diseased eyes. And these are the results. So what about the reliability of different readers in analyzing the FIZ? Well, this was extremely high. I mean, the correlation, interclass correlation was extremely high for all 3 readers, especially for more superficial layers. This was definitely lower for the deep capillary plexus, as you can see here, at higher field of view with less resolution. On the other hand, vessel density was excellent. I mean, all readers were able to evaluate in the same manner. Doesn't matter about the size of the cube that were investigated, in the same manner the vessels. The other question was then about the agreement between the different plexuses. So in other sense, we wanted to test if we look at some vessels, let's say in the superficial capillary plexus in 3x3, is that exactly the same that we can find in the 6x6 or in the 12x12? Well, unfortunately, this was not the case. So in a sense, it seems that we cannot compare different sites of cube of acquisition of OCT-A, even with swept source. And so repeatability is only true for the same sites of analysis. We cannot compare 3x3. What are the outputs, quantitative analysis of 3x3 with a 6x6 or with a 12x12? And this is even, as you can see here, much more important for disease eyes, and particularly because in some eyes with diabetic retinopathy, there was a loss at the level of the deep capillary plexus of vessels, and the FAZ exceeded the field of view, the 3x3. So the next question was for us, okay, swept source seems to be superior, especially for the choroid and the choriocapillary. So is that comparable what we see with the spectral domain OCT when we look at the same patient, the same lesion, by using a swept source OCT? And then we looked at patients with choroidal neovascularization, so with AMD. These are the results. As you can see, the area of a choroidal neovascularization was exactly the same, looking both at spectral domain and swept source. But what was striking was about vessels density. So it seems that there is a higher density and significantly higher density of vessels in the context of choroidal neovascularization if we use a swept source OCT. So it seems to be true. Swept source OCT is particularly useful when we look at the fine details, tiny details in the context of at least diseased eyes. In a sense, let's try to follow patients with the choroidal neovascularization. We want to test the possibility to make some regression of tiny vessels. Well, performance of swept source OCT seems to be superior than spectral domain OCT. And the other question was, okay, so it seems to be superior than spectral domain, but are all swept source OCT the same when we evaluate a choroidal neovascularization? You know that there is only one swept source OCT in the market, but there is another one that is a Melier research tool, and we try to compare these two images, and again the results were striking and somehow also difficult to interpret. When we look just at the size of the lesion, well, this was the same looking both at 3x3 and 6x6. So no doubt the two machines, the two outputs are comparable. But when we looked at the vessel density, and so this is the most important thing that we want to see in our patients, especially patients that are being treated, well, there was some difference. But this difference seemed to favor one machine in terms of vessels density when we looked at the 3x3, and the other machine looking still at vessel density when we looked with the other machine. So this is the very first study in that sense, so it's difficult to interpret these results. So in conclusion, when we look at the FAZ, this is a very repeatable evaluation. It doesn't matter from the operator, it doesn't matter from the field of view that we are using in our patients, so it's very repeatable evaluation in our patients. On the other hand, vessel density still represents a problem in the possibility to quantify and follow patients. And this because while in the superficial capillary plexus there is a high repeatability, at least among the readers, while the quantification varies depending from the size of field of view. So we cannot compare different cubes of OCT-A at this time. What about web source OCT in general? Well, this seems to be superior to spectral domain OCT-A in terms of visualization of fine details of the context of choroidal neovascularization. And what about agreement between web source machines? Well, for sure, we have a high resolution in the context of visualization of CNV, but the results, depending from the sites, are still doubtful, so further studies are needed. Thank you very much for your attention, and thanks to my coworkers.